This is Imamul Kabir. I'm from Fluidic Device Lab on Department of Mechanical Engineering on Cook University. Here I'm going to present a microfluidic device which is used for cell pairing, fusion, and cell culture. And I also suggest some its necessary improvements. Here is the outlines of my presentation. First, device fabrication, operation of the device, and its application in cell culture. Then I'm going to suggest some new methods for its improvement. Here is the device fabrication. In this microfluidic device, there is two portions. One is upper parts and there is lower parts. The upper parts is used for the cell culture and lower parts is used for cell trapping. And in the lower part, there is cell trapping, fusion, and pairing occurs. In the figure C, we can see the details of the manufacturing process of this device with the soft lithography. First, for the two portion, there is a common of the glass as a substrate. In the first portion, for the lower part, the gold electrodes are deposited on the glass substrate through a standard lift-off process. Then a 2 micrometer thick layer of BN30330 photoresist was spin coated, and this layer will act to protect the cells. Over it, there is another negative photoresist of 25 micrometer thick layer. This is all about the lower part, and on the another part, the upper part, first on the glass substrate, there will be another negative photoresist of SU8, but there is a rather big microchannel, and on the negative photoresist, we have to power the PDMS mixture 10:1 by weight PDMS and curing agent. Silgard 185 Dow burning was used as a PDMS agent, curing agent for this mold. Then, after curing at 80 degrees Celsius 30 minutes, the PDMS layer was peeled off, which we can see in the figure C, and bonded to the electrode photoresist structure, which was made first. Then, to enhance the bonding between these two structures, oxygen plasma treatment was used. And in the figure B, we can see the details view of the two well of this device, and the whole device we can see in figure D. In the device operation process, there is three functions of this device. First, the cell trapping, then pairing, and fusion. For this operation, first, positive dielectrophoresis force is activated through the electrode structure. So the cell will be loaded at the trenches of the electrodes, but they will be not in a good contact. For this, for making this good contact, after the DFE buffer solution washing, then the negative dielectrophoresis with a low frequency comparatively, the positive dielectrophoresis was applied for good contact of the cells. After the cell attachment, the cell fusion occurs, and the fusion can be occurred by two processes, electrical pulses or the PAG solution with the buffer. And after the fusion, the device is gently flipping to transfer the cells from the capture well to the culture well. After the cells fall into the culture wells, the entire device will be placed into the cell culture incubator at 37 degrees Celsius for several days of cell culture and observation. And this is the application of the device in the cell culture. In the first figure, we can see the cells are arranged in the cell culture medium at a very little, and they have a good distance between each other. But after 36 hours, the cells are growing rapidly. But after a long-term duration, 72 hours, we can easily see that cell migration occurs and the proliferation of the cell is much. So these results can evidence that the fused cells are viable, and we can use this microfluidic device integratedly for the three operations of trapping, pairing, fusion, along with the cell culture. With this microfluidic device, I'm going to suggest three methods mainly. The first method is to incorporating new materials before the cell-cell attachment. This is polyethylene glycol, PEG lipids polymer for the cell surface modification before the cell-cell attachment. After this attachment with the conventional PEG-induced method, we can easily increase the cell fusion efficiency. And here, this is the chemical structure of the PEG lipids oligopeptide polymer. We can easily see that with these materials, with the proper concentration, the attachment area is increasing much at the left figure, and at the right figure, we can easily see that apart from the only fusion or only attachment and fusion efficiency is much higher in terms of cell fusion. So this paper suggests that the efficiency can be increased up to 64% from the 8.4% with the conventional PEG-induced method after incorporating these materials on the cell surface. And this is the method 2. Here I'm going to suggest that materials, heparin sulfate, which absence significantly increases the cell-cell fusion efficiency. So to remove the heparin sulfate from the surface, I suggested two materials, heparinase 2 and heparinase 3, to clip edges from the cell surface. And the figures left and right, the luminosity, actually the luminosity means cell fusion efficiency with these materials in the left figure is increasing for the 3 cells respectively. And on the other side, for the 2 cells, HeLa and Vero, with much concentration of these materials, the cell fusion efficiency is increasing rapidly. In the third methods, I'm going to express the bipolar pulse in terms of unipolar pulse. Here in this paper, the unipolar pulses, that means the DC voltage is used, but this unipolar pulse is responsible for high mortality of cell, and cell fusion rate is also restricted by this cell mortality. So I can easily suggest in this method to incorporate bipolar pulses that cannot only reduce the cell mortality remarkably, but also improve the cell fusion efficiency. The figure, the fusion rate, you can easily see that the fusion rate for the bipolar pulses is increasing comparatively to unipolar pulses, and the mortality rate is decreasing so rapidly comparatively to the unipolar pulses. So this is the important improvement method to use the bipolar pulses instead of unipolar pulses. Here is the references of my presentation. Thank you for watching for my videos.